If you want to understand why endotoxin testing of pharma is so important, a cluster of endotoxin poisoning cases that occurred in Australia provide a poignant example. In 2015, a 41-year-old patient who, that day, had received an infusion from a compounding pharmacy turned up in an ER room in Sydney. She had a fever, muscle twitches, vomiting, and pain in her abdomen, neck, and back. She was treated with broad-spectrum antimicrobial therapy and remained hospitalized for a week. Then, 2 weeks after being admitted, she developed a complication of the antibiotics, C. diff, an excruciating inflammatory condition in the colon that can sometimes be fatal. Ultimately, health authorities linked a total of 7 cases of illnesses to that pharmacy she had visited, and they noted that there were several lapses in aseptic production at the site, and that a powder used to make the medication she was given contained high levels of endotoxins. I'm Megan Parrish, and you're listening to a special Solution Spotlight edition of Off Script, Pharma Manufacturing's podcast that goes beyond the pages of our magazine to discuss the issues that matter to the industry most. In this episode, we're taking a close look at a platform aimed at providing an easier way to conduct endotoxin testing in pharma facilities. Called the Sievers Eclipse Bacterial Endotoxins Testing platform, the solution, which is made by Suez, includes a number of features that deliver both automation and compliance. Using groundbreaking technology, the Eclipse decreases the needed pipetting steps and is easy to install, reducing setup time by a whopping 85%. It also reduces the use of the required LAL reagent by 90%, which helps companies reach sustainability goals. So, to learn more about the Eclipse, I'm joined today by Hayden Skalski, the Life Sciences Product Application Specialist for the Sievers line of analytical instruments at Suez. Hayden has over 8 years of experience in the pharma industry and in quality control microbiology, and he's presented on numerous topics surrounding endotoxin testing. All right, Hayden, thank you so much for joining me.
Hey Megan, thanks for having me.
Absolutely. Let's start by taking a look at the landscape for endotoxin tests. What are some of the options for endotoxin testing today, and how do they compare?
So currently there are 3 types of approved endotoxin testing. One is the gel clot method and then 2 photometric methods. Gel clot was the first LAL test that was approved back in 1977. And to simplify that, how this method is performed, the LAL reagent is added to a test tube which contains a sample, and then that is incubated for an hour at 37 degrees. Upon that incubation completion, the tubes are inverted, and if the liquid has gelled or clotted, that would indicate the sample is contaminated with endotoxin. If no gel has formulated, then no endotoxin is present. This is a pass or fail test, so you either get a positive or a negative test, and that this is considered a qualitative test, so it's either passing or failing. The 2 photometric methods, which are more automated and give you a quantitative result, so you get an actual numeric result, are the kinetic chromogenic method and the kinetic turbidimetric, which uses a spectrophotometer or a absorbance reader to perform the assay. The turbidimetric method measures an increase in turbidity, or the time taken to reach a particular level of turbidity or onset time. The chromogenic method measures a color change, which would be yellow over time. So this kind of happens when a colorless chromogenic substrate is added to the LAL reagent that causes the yellow color to develop after a cleavage of the chromophore, which is known as para-nitroaniline or PNA. So the more endotoxin that is present, the more yellow the solution will become. And generally, the turbidimetric and chromogenic techniques are run in the 96 well plate format, and they are very time-consuming tasks. And there are newer technologies that have been developed to run these LAL tests, and those are a cartridge-based technology and a microfluidic plate, such as the Sievers Eclipse. There are also some recombinant assays out there, which uses a synthetic form of factor C instead of actual blood cells that is found in the horseshoe crab's amoebocytes that is responsible for the enzymatic reactions to detect endotoxin. And this type of technology uses a fluorescence reader, which is separate from the absorbance readers used in traditional LAL methods. This method, however, is not compendial, and it requires to undergo a alternative method validation per USP 1223.
Yeah, and I was just about to ask you about some of the challenges and limitations associated with these different methods. Can you talk a little bit about that and tell me why the industry is looking for improvements on endotoxin testing?
Yeah, so there are many reasons why improving the LAL test is so beneficial and why the industry is striving to improve it. So the bacterial endotoxin test is a critical release test, meaning these life-changing medicines and these products cannot be released to the public until it passes the LAL test. However, with the traditional methods like I described earlier, such as gel clot and the 96-well plate methods, come some caveats, and these older techniques are extremely manual and they require a significant amount of time to set up these tests. So a lot of the analysts in the labs will spend most of their day kind of setting up and running these older techniques. So I have to say, the more human intervention, the more likely the LAL test is prone to errors. And we have to remember that the LAL test is a very sensitive test, detecting down to 1 parts per trillion. So that's equivalent to a grain of sand in an Olympic-sized pool. That is pretty remarkable if you think about it. However, that also means any type of small contamination by the user will most likely be detected. So companies, want to get their product out to market faster to help these patients. But if there are failed or invalid LAL tests, this means a lot of retesting, a lot of investigations, which means product is getting held up. So by adopting newer technologies that are currently available today, such as the Eclipse microplate, the QC labs can be assured that not only will it significantly reduce analyst hands-on time, but it will also reduce the percentage of potential invalid tests and time spent retesting and writing all of these investigations. And the time not spent doing those 2 tasks will also free up analysts to run more LAL tests and focus on other lab-specific tasks. And I'd like to just talk about kind of another reason why this industry seeks to improve LAL testing, and that is data integrity. How this can be tied back into human error, and that a lot of these processes tied into LAL testing allow for this error to occur. So as labs use more dated methods, such as the gel clot, they are subjecting themselves to these data integrity issues and a lot of human error due to the manual processes of the test. The gel clot test, like I just mentioned, it's subjective, meaning it's based on an analyst's observations. And this observation needs to be verified by a second analyst, which they may interpret the results differently. And as kinetic methods are introduced and as newer technologies are introduced, the subjective aspect goes away, and the new data software that is tied into these newer methods, they interpolate the data, which ties back to the software and is manually read by the analyst. And this ties into the 21 CFR Part 11, so maintaining that data integrity compliance.
Yeah, so lots of ways that the method can be improved upon and lots of ways this technology really helps. So let's say that a company decides that they're going to go ahead and implement a new solution for LAL testing or switch from one solution to another. What do you recommend that they do in order to go ahead and initiate that kind of change?
Yeah, so I can start off with the easiest change that a lab would be looking to do. That would be going from the same method to the same method. So example here is if you're running a chromogenic 96-well plate and you want to switch to the chromogenic method utilizing the Sievers Eclipse microplate, this is the easiest because you're going from chromogenic to chromogenic, and that pretty much means the biochemistry is still the same and still chromogenic. So that is going to be the easiest switch. And that is recommended to do that is a side-by-side study of the current platform you're using with your samples and using the new validated platform that you want to switch to ensure the product is suitable. And since you are using the same biochemistry in the chromogenic method, a 1-lot validation would be sufficient at the same dilution if the results are comparable. Another example is if you are currently testing a gel clot method or kinetic turbidimetric and you're looking to switch to kinetic chromogenic using the Eclipse platform, This requires a little more of a comprehensive approach, but it's still easily attainable. So what you want to do in this situation is a method suitability test. And this basically means you're going to perform this suitability test, which is also known as a inhibition enhancement screen at different dilutions to show that the products can be accurately tested and recover endotoxin using the new method and that there is no interference. So once a suitable dilution is chosen using the new technique, you will perform a 3-lot revalidation of the product. I like to say it all doesn't have to get done at once. Labs can pick and choose at their convenience when they do this revalidation to make it more suitable and time worthy for them. And when you're changing methods or you're changing lysate vendors or you're switching platforms for a drug product, QC labs often think it's a daunting task and a complicated process due to the regulatory filings. So it is a recommendation when including your product in a filing for endotoxin to state briefly how it is tested. So an example is you would state drug XYZ utilizes one of the compendial photometric kinetic methods for LEL testing. That way, if you're ever switching vendors or methods, it makes it easier to change and you're not tied into one specific vendor or specific method. Now, there is some guidance on how to go about changing a filing, and it is a guidance for the industry by the FDA called Changes to an Improved NDA or ANDA. Then there are 2 types of filing changes, and that is a moderate change and a minor change. So a change from the gel clot method to a turbidimetric or chromogenic method would fall under a moderate change known as CBE 30 or CBE 0, and the CBE stands for changes being effective. The CBE 30 requires a submission of a supplement to the FDA at least 30 days before the distribution of the drug product made using the change. A CBE 0 would be considered as going from a 96-well plate chromogenic assay to the Eclipse chromogenic assay product. Now, for this CBE 0, the FDA may identify certain moderate changes for which distribution can occur when the FDA receives a supplement. So, in other words, the FDA will approve the LEL changes as long as the new method is compliant with USP 85. Now, an annual report is the last filing change and is considered a minor change. A company will submit an annual report to the FDA telling them of the changes that were made. So switching lysate vendors would fall under this category, and going from a 96-well plate KCA to the Eclipse KCA product can also be considered in an annual report as well. So a lot of companies and their quality assurance or the regulatory affairs department will have their own internal requirements for going about how to do a filing change, and their filing change could be a CBE 30 for LEL vendor change compared to doing an annual report. So it all depends on the internal specifications for your company.
Okay, great. And I was going to ask you about system validation. You touched on this a little bit, but I wondered if you wanted to add anything. So tell me a little bit about some of the important aspects of system validation in terms of LAL testing.
Bacterial endotoxin testing platforms, so that they must be validated to demonstrate that a valid endotoxin test can be carried out on the substances or products concerned. Now, this may entail a procedure for removing interference, and you want a system that is fully compliant with 21 CFR Part 11 and data integrity guidelines that align with Alcoa Plus to ensure there's no doubt with regards to accurately generating and maintaining that data. You also want to ensure that the system can demonstrate linearity, accuracy, precision, sensitivity, equivalency, and a 1-to-1 lysate to sample ratio. Now, this will all fall under the IQ, OQ, PQ system validation, and this is extremely important, to successfully carry out these validation requirements.
Great. And of course, sustainability is a huge topic right now. In fact, sustainability used to be one of those things that businesses worked into their strategy if they could, but now it's kind of a given that companies have pretty robust sustainability programs. So tell me how this fits into sustainability goals, and what do you think is important in regards to endotoxin testing and sustainability?
Of course. So I like to just start, by saying, you can thank a horseshoe crab if you've ever received the recent COVID vaccine or an IV treatment or any injectable drug or medical device. These products, they cannot be sent out to market until they've been tested for bacterial endotoxins, which if they were, sent out in the market and they were contaminated, it would cause a fever, adverse reactions, and even death in a patient. So very important for these products to be tested for endotoxin. Now, the horseshoe crab blood contains factors that, if extracted, could be used to manufacture what we call LEL. So that stands for Limulus amoebocyte lysate. And that's an aqueous in vitro reagent that holds the potential to significantly improve our ability to whether injectable drugs have been contaminated with endotoxin. Now, the vast majority of this reagent comes from the North American horseshoe crab, Limulus polyphemus, which is found up and down the eastern seaboard of the United States. So as vaccines ramp up worldwide and new treatments for diseases emerge, there's an increasing demand for the horseshoe crab's unique blood. Now, there are sustainability initiatives and even laws enacted to protect the crab, so they cannot be used for bait or fishery practices, and they're in place so they can only be used for biomedical purposes, such as harvesting them for producing this important reagent. However, there is a still high probability that these species become vulnerable to baiting and habitat loss, which could lead to a decline in population. As such, it's very critical that we find ways to conserve and optimize the use of this resource as much as possible. So instead of traditional LEL tests such as gel clot and 96 bulk plate assays that I touched on earlier, there are newer technologies such as the Eclipse analyzer that now offer LEL testing using up to 90% less LEL reagent than those traditional methods. It's vital that QC labs pursue newer automated methods that use less reagent. Microfluidic technology that utilizes small volumes of sample and reagents in small spaces will help conserve this Limulus species for years to come, as well as continued monitoring of the horseshoe crab to protect it. So life-saving medicine does depend on this crab and the reagent that it produces. And in the industry as a whole, we must work together on conserving this special creature by looking to newer technologies that utilize less reagent.
Now, I'm going to circle you back to the topic of validation. I was wondering if you could tell me, why is it important to perform product validation?
Sure. So product validation, this is also known as product screening, or some of us like to call it the inhibition enhancement screening. And this is performed on new products or existing products that are going to be changing LEL methods. So this typically involves 3 lots of product, and these are screened at different dilutions to see which dilution is non-interfering and gives the best spike recovery. And the best spike recovery is a deal to be around 100%. So the product validation pretty much or basically gives validity to the test, ensuring the product is suitable at that dilution and it can recover endotoxin accurately. Now, most products, aside from water, will interfere with the LEL test, so it's very important to do this type of screening. Unfortunately, the LEL assay is very sensitive, and most often than not, if you do have interference, it can be overcome with simple dilution. So the inhibition enhancement screening is just that. You're screening the product to see if it interferes with the LEL reagent that will inhibit endotoxin recovery less than 50%, because in the LEL test, we have a range of 50 to 200%, and we want to be inside that range. So if you're less than 50%, that's going to inhibit the LEL reagent that's interacting with your product. And we also want to see if it's going to enhance the recovery, so greater than 200%. Now, the main concern with inhibition is it could underestimate the value of endotoxin, and the product could potentially be released to market. So we call it a false negative. So if there's something inhibiting that reaction, it might give you a passing test, which in real time it's not. It's interfering. So we call that a false negative, and that could go out to the market and be released, and that would be a big problem. And now enhancement is much less dangerous than inhibition, because as the product will not be released to the market, it'll just show levels of recovered endotoxin. And nevertheless, the screening is done during the validation stage and must pass all criteria outlined in Chapter 85 of the USP before being validated. So you'll get to see during that screening if something's being inhibited or enhanced.
Okay, great. And I want to talk a little bit more about the technology that is behind this platform, the microfluidic technology. Can you tell me what some of the benefits are of that, and also just a bit more about how it works?
Sure. So Microfluidics in endotoxin testing is a microfluidic liquid handling that facilitates accurate and rapid dispersion of the reagents and samples with drastically reduced volumes of sample and LEL reagent within the Eclipse microplate. So it's achieved by using all these little microfluidic channels and metering chambers, and it uses centrifugal force. So the microplate spins really fast to control and automate all liquid measurements, flows, and mixing and preparation for analysis. And microfluidic technology is automation in the simplest way. So it simplifies endotoxin testing in a platform that is extremely easy to set up and use without the need for robotics. All standard curves and positive product controls, or PPCs, are embedded on the microplate for ease of use and comes with significantly less pipetting steps than traditional tests. In addition, the microfluidic liquid handling precisely measures all liquids for the end user, and that means basically that the precision typically required for physical action of pipetting is eliminated through the precise design of the Eclipse microfluidic microplate.
Okay, great. And at the beginning of this conversation, we talked a bit about the benefits of platform technologies like the Eclipse and some of the ways in which they're improving endotoxin testing. So I want to wrap up by asking you to elaborate on that a little bit more. And in particular, I was wondering if you can tell me how the Eclipse reduces the potential for human error and also improves data integrity.
Absolutely. The Eclipse microplate comes preloaded with embedded standard curves ranging from 50 EU per mil down to 0.005 EU per mil from reference standard endotoxin, or RSE. And it also contains positive product controls and duplicate up to 21 samples. So you don't have to rely on an analyst creating a standard curve every assay. By doing that, you could have consequences. If the curve they generate is too potent or too weak, and that could have a devastating effect on your test. So the Eclipse microplate takes care of that for you by having everything embedded. So that basically, by having that already set on the plate, you're already removing potential for a lot of human error. Now with traditional 96 well plate assays, every sample is spiked in duplicate manually. So any small distraction, could lead to a misspiked well, and that leads to an invalid sample and then an investigation. So we take that out of the picture too. So that really leaves little room for error. Now also with traditional plate assays, if you do a full plate of 21 samples, that is a total of 242 pipetting steps for a full plate. With the Eclipse microplate, there are 27 pipetting steps. So this reduction in pipetting steps not only reduces the risk for human error enormously, but it also reduces the risk for repetitive injury, and it reduces a lot of time spent executing the test. And kind of just to wrap that part up, by removing so many aspects of human intervention, the Eclipse drastically improves, data integrity on that end. And just speaking, a little more on data integrity, the Eclipse platform comes with the Eclipse software to analyze endotoxin. And the Eclipse software offers a full 21 CFR Part 11 data integrity compliant package that includes Alcoa Plus principles and adheres to those guidelines. So, when an assay is running, it transmits data to the software every 5 seconds. So you can go check progress frequently throughout the assay to see how it's doing and see the data in real time. The software also has a highly customizable permission and product accessories database in which a lab supervisor can set up different analysts to test only certain products or samples and give different permissions. So it's really up to the lab how they want to set it up, but at least we give them all types of accesses, and they get to pick and choose how they want to run and set that up. And just to kind of conclude it, the software it has routine database backup and restore, so data will not be lost if
You've
there
been
was
listening to a special
disaster
Solution
to
Spotlight
occur or
edition of
something
Off
shut
Script.
down the system.
Stay
Everything
safe,
will
everyone, and be
maintained.
well.
So it really ties back into to be very, very data integrity compliant overall.
Well, that's great. A lot of great improvements with the Eclipse software, it sounds like, or the Eclipse platform, rather. Thank you so much for taking the time today to explain why this is such an important technology and why the issue of endotoxin testing is so critical for the pharma industry.
Yeah, thank you so much for having me on, and I was happy and excited to discuss all these important questions surrounding endotoxin testing.
Yeah, of course.